Dear Outrun the Sun team, supporters and friends. My name is Christian Posch. I'm the 2014 Outrun the Sun Research Scholar. And back in 2014, I've been studying the role of PD-1 on tumor cells together with another Outrun the Sun Research Scholar, Professor Tobias Schatton at Harvard University. PD-1 is a so-called immune checkpoint molecule, and one can think of it as like a negative regulator that puts a brake on immune cell activity after their job is completed. You can imagine that this regulation is critical for everybody, otherwise we would live in a state of permanent inflammation, and that would obviously be a bad thing. The problem is, however, that tumor cells are able to use this mechanism to hide from immune cells. And that is critical because the job of immune cells is not only to clear our bodies from infections and pathogens, but also from tumor cells. So if a tumor cell is able to escape immune surveillance, it gives them a survival advantage. In our research, we were able to show that this important immune checkpoint molecule, PD-1, can be found not only on immune cells, but also on tumor cells. This was quite controversial at the time, because it was believed that immune checkpoint molecules are only to be found on immune cells. Today we know that PD-1 has an important role on tumor cells too, and targeting this receptor can contribute to help improve melanoma treatment. In 2015, we were able to publish our findings in the prestigious journal Cell, and I'm very happy to send anyone a copy if you want to read a little more about it. Your support helped me tremendously in completing this study and share our results with the rest of the world. After my time as a research fellow at the Brigham and Women's Hospital in Boston, I moved back to Europe, and in 2018 I was appointed head of Skin Cancer Research Center here at the Technical University of Munich. We treat all different kinds of skin cancers, but melanoma remains one of our biggest challenges. This is why my research team and I work on several, mainly translational research topics that aim to further improving treatment options for melanoma patients. In this context, I believe that there is much to be learned from looking at the very start of melanoma genesis. Wouldn't it be great if we could prevent the majority of melanomas before they even become a problem, before we even have to use fancy, sophisticated treatments? My model system for studying melanoma genesis is the so-called nevus-associated melanoma. There are melanomas, these are melanomas that arise in pre-existing moles that did not change for decades, for example, but all of a sudden start to give rise to a melanoma. This kind of melanoma makes up about 30% of all cases. The other 60% are so-called de novo melanomas and develop on previously unremarkable skin. And there is a very small group that develops on tissues that are probably representing a very different kind of melanoma altogether, like mucosal melanomas or uveal melanomas. Today we understand that melanoma is just not one disease, but it's many different types of diseases. And we're only starting to distinguish and comprehend all the different types of melanomas with sometimes very different biological trajectories. What we're seeing in these nevus-associated melanomas is that a fundamental mechanism that protects cells from turning cancerous after genetic insult, like an oncogenic mutation, is bypassed. This protective mechanism is called senescence. Some of you now might say, well, wait a second, senescence is not a good thing. And generally, I would agree with you if we are talking about the context in normal cells. Here, senescence can be a major driver of tissue and organ aging, frailty, and also disease. However, sending cells into senescence can also be a good thing if it means to stop a cell from uncontrolled growth. This type of senescence, so-called oncogene-induced senescence, is very powerful, but in some cases, as you guessed it, like in nevus-associated melanomas, this protective mechanism fails and cells escape senescence programs. My lab is now trying to understand this process of senescence escape with the goal to get new insights into the biology of early melanoma development. I hope that our research will open up new avenues for melanoma prevention and also melanoma treatment. And I'm very happy to update you on our progress in the future. For now, I would like to thank you again for your support, for your voluntary work, for your Out Run the Sun runs, and all your campaigns and activities. I think this is just wonderful and highlights that we can achieve a lot when we all work together and trying to keep our skin safe and healthy. So keep up the good work. We'll do the same here, and I hope to see you soon at one of the Out Run the Sun events. Cheers.
Hi, my name is Hiroshi. I'm a research assistant professor in the Department of Physiology and Biophysics at Boston University School of Medicine. Although previously I was a practicing dermatologist, currently I'm a full-time researcher interested in the molecular mechanism of melanoma metastasis. I'd like to thank Outlander Sun for funding my research and for giving me this opportunity to share with you my melanoma research. As many of you have noticed, black cats and dogs frequently have white feet and white belly spot. These black colors are produced by melanocytes, pigment cells responsible for the coloring of our hair and skin. White belly spots are caused by a delay in melanoblast migration during embryogenesis. Melanoblasts are precursors of melanocytes and migrate long distances during body development as shown by arrows in this picture. The feet and belly are the farthest destination for this migration, so a delay causes blank area of melanocytes. We found that mice that do not express the motor protein myosin 10 have white belly spot, evidence that myosin 10 is necessary for melanocyte migration. But why are white belly spots important for melanoma research? Melanoma frequently metastasizes, that is, the cancer cells spread from the place where they first formed to other parts of the body. This is a major cause of death in melanoma patients. Our general concept is that melanoma metastasis uses the same mechanism as melanoblast migration in embryos. Thus, we speculate that our gene of interest, myosin 10, controls not only melanoblast migration but also melanoma metastasis. I'll introduce a little bit about myosin 10. This is a molecular structure of myosin 10. Myosin 10 interacts with actin filament with its motor domain, which converts the chemical energy of ATP to mechanical work, resulting in its movement along actin filaments by a hand-over-hand mechanism. And the tail domain binds to specific molecules. Thus, myosin 10 functions as a transporter in cells. Using actin filaments as tracks, it transports specific cargo molecules to specific intracellular sites. So what happens in melanoblast cells after myosin 10 depletion? This is a representative image of cultured melanoblast cells. Many control cells show long pseudopod-like protrusions. In myosin 10 depleted cells, long protrusions are reduced, and migration speed is slower than that of control cells. Then the next question is, what happens if myosin 10 is depleted in melanoma-inducible mice? As we speculated, we found that myosin 10 depletion in melanoma-inducible mice significantly decreased melanoma metastasis. Thus, myosin 10 plays important roles not only in melanoblast migration but also in melanoma metastasis. Surprisingly, myosin 10 depletion also delayed the onset and development of melanoma. We also found that myosin 10 is highly expressed in human melanoma samples, and increased expression of myosin 10 decreased patient survival. However, little is known why and how myosin 10 depletion inhibits melanoma development and metastasis. The Outlander Sun Scholar Award will give us an opportunity to investigate this mechanism in depth. For example, what cargoes of myosin 10 are responsible for melanoma progression, and how does myosin 10 work in other cell types such as immune, stroma, and serial cells for melanoma progression. By accomplishing this project, we hope to gain insight into how to prevent melanoma progression, resulting in the development of new types of drugs for melanoma treatment. Thank you.
Hello everyone. It is an honor to be the recipient of Outrun the Sun. I am Poon, a postdoctoral fellow in the Center for Immunotherapy and Precision Immuno-Oncology at the Cleveland Clinic, Ohio. I graduated with first-class honors for my bachelor's in industrial chemistry and went on to earn a Master's of Science in Bioengineering and a PhD in Clinical Oncology. After my first postdoctoral training program at the Karolinska Institute in Stockholm, Sweden, I joined Dr. Brian Gassman's laboratory at the end of 2018. My research focuses mainly on oncology, with a specific interest in the discovery of predictive biomarkers for immunotherapy resistance. As known, melanoma is one of the deadliest skin cancer. Despite the massive improvements in melanoma survival, it is disheartening that half of these patients still fail to benefit from immunotherapy, necessitating an early and prompt prediction for treatment outcomes. The fact that the incidence of melanoma continues to increase, eclipsing 100,000 cases per year in the United States in 2020, make it even more pressing to unravel immunotherapy resistance in melanoma. The question is, how do we overcome this problem? Leveraging on our large volume tumor specimens from surgical resections, we are able to investigate the heterogeneity and complexity of the interaction between the tumor and the host immune system. This approach should better allow us to identify melanoma immunotherapy resistance, helping to tailor the best possible treatment modality for patients. To understand the heterogeneity of CD8 T cells, a subset of immune cells that are critical in immunotherapy effectiveness, we perform single-cell RNA sequencing analysis on matching immune cells from tumor and blood of melanoma patients. We discover a distinctive subset of dysfunctional CD8 T cells that are not only highly heterogeneous, but also metabolically active. Most importantly, this key finding leads us to identify a unique gene expression profile predicting for immunotherapy responses in both the tumor and peripheral blood. It is known that one of the major challenges in patient care management is the ability to administer the right treatment modalities with sustainable outcomes. To do so, we need reliable predictive biomarkers and tools to correctly predict for immunotherapy response, which remain a pressing challenge in the field. Although research can provide a broad understanding of how to treat each patient, ultimately, the clinician determines which regimen is ideal for individual based on the patient's own therapy responses. Therefore, it is vital to overcome the issue of predicting therapy responses in faster and more effective ways by using minimally invasive approaches. However, to date, cost-effective and reliable predictive biomarker platforms for immunotherapy response are still largely underdeveloped. Despite excessive efforts to solve this hurdle using various different technologies, there is no standard correlative assay used for immunotherapy in the treatment of melanoma. In an attempt to overcome this challenge, our group will examine our newly discovered fingerprint of CD8 T cells predicting for immunotherapy resistance in melanoma patients. We aim to develop a less invasive blood-based predictive tool by integrating two different techniques, the flow cytometry and RNA hybridization. The development of minimally invasive blood-based predictive tools will greatly enhance the immunotherapy response prediction rate. Our long-range objective is to utilize these blood-based predictive tools to improve prediction and treatment decision-making for melanoma patients, and ultimately in any cancer patient receiving immunotherapy. We believe our work holds great promise for precision and personalized immunotherapy by maximizing benefits and improving patient care management. Hence, the support from Outrun the Sun not only has a very important impact on my current research, but it holds great potential in improving melanoma patient care. I am truly indebted and grateful for the support and wanted to take this opportunity to express my gratitude to Outrun the Sun for believing in our research project. Thank you very much.